Hello everyone, welcome to the cell biology series of Biologics Everything Biological. So today we will study the lipids of plasma membrane. So first see the cell membrane syllabus for the CSIR NET. So here you can see that the membrane structure and function contains some very important part which are you will get every year 4 to 5 questions from this part, that is structure of model membrane, lipid bilayer and membrane protein diffusion and the transport mechanisms. So here in this plasma membrane series we will cover all these parts in our subsequent lectures. So let's start with lipid bilayer. As we all know that lipids bilayer is a very important part of the membrane. So the Singer and Nicholson model of plasma membrane says that the lipid, protein and lipid, these three are the major components of plasma membrane. So to start with, how lipid reacts when it comes in contact to water. So if you think that these cones are lipid molecules, are single lipid molecules, what happens when it comes in presence of water? This cones turns and comes together and form a lipid micelle. These micelles are actually very important for the transport of drugs in case of different drugs in humans. And what happens, how this lipid pact and lipid bilayer forms when these types of phospholipids they comes together, they forms these kinds of a planar structure. So this is how the lipid bilayers forms. The hydrophobic tails comes together and these hydrophilic heads are actually comes in presence of the water and they form a sheet like this. But this planar structure is energetically unfavorable. So what that does is that they seal the compartment to form a phospholipid bilayer. So in maximum cell we'll find that the cell structure is kind of liposomic or kind of spherical. So let's start with liposome and black membrane. So you know that the understanding of lipid bilayer is done artificially outside the cell. So when we study the membrane and its function outside, we make a spherical structure of lipid bilayer which is known as liposome. These liposomes are very much used as a drug transporter. Now here you can see this is the planar structure. It is actually help in the study of the solute transport from one solvent to another solvent. So why it is called black membrane? This planar structure, when put in a small hole between two solution, it looks black. Because of that, it is known as the black membrane. And these black membranes are used to study the soluble molecules of the semi-permeable membrane, the solute transport of the soluble membrane, semi-solid membrane. So now, how this lipid bilayers behaves in cell? So these lipid bilayers are not static; they have a kind of movement inside the membrane. So this lipid bilayers diffusion, this types of lateral, this side by side diffusion is known as lateral diffusion, and from flip flop diffusion also these do, and they also rotate among themselves. So these are the three kinds of movements which are specifically observed in lipid bilayers. So enzymes involved in these processes are flippase and floppase. Another is scramblase. So from here to here, this transport is sometimes energetically unfavorable. So it requires enzymes known as flippase and floppase. So what flippase does? Flippase takes the lipid from the exoplasmic site to the cytoplasmic site and floppase to the reverse, and both are ATP dependent transporters. The scramblase on the other side is not a ATP dependent transporter. What it does, it is a energy independent transporter, and it does not require any enzymes for the any ATP to transport from one side to another side. So this is a very important part for the CSIR net, and you read everything which I have written here is important for the CSIR net question. Now, as we know that the phospholipids are one of the major component of plasma membrane, but other than phospholipid, there are many other lipids which are responsible for the formation of the plasma membrane in both animal, plants, and bacteria. So what happened here is that you see the other than phospholipids, cholesterol, glycolipids, and sphingomyelins are also important part of plasma membrane. Now, the phospholipid and cholesterols are present everywhere, the outer leaflet and the inner leaflet both. But here you have to see that in the outer leaflet, sphingomyelin, glycolipids are mainly present in outer leaflet. They are not present mainly in the inner leaflet. Those sphingomyelins are present, but glycolipids are not present in the inner leaflet of the cell membrane. Another thing is that the phospholipids which are present outside are mainly phosphatidylcholine. The phosphatidylserine, phosphatidyl'inositol, and phosphatidylethanolamine are mainly present. Phosphatidylserine and phosphatidyl'inositol are mainly present inside the membrane. So this is very important because there will be some questions, trick questions, where we will find that the phosphatidylserine or phosphatidyl'inositol, they will ask that which side of the membrane they are present, inner leaflet or outer leaflet. So you have to remember that phosphatidylserine and phosphatidyl'inositol are present in the inner leaflet of the cell membrane. This is a very important part. From here you will find 1 or 2 questions almost every year. Now, structure of different phospholipid. How the phospholipid looks? Phospholipids looks like that there is a phosphate, the glycerol, and the long hydrocarbon chain. This hydrocarbon tail is actually hydrophobic, and above all there are some group present. If it is choline, then it will form phosphatidylcholine. If it is serine, then it will form phosphatidylserine, and if it is ethanolamine, then it will form phosphatidylethanolamine. Another thing is that the sphingolipids are also phospholipids, but here other than glycerol, the sphingosine is present. So you have to remember that the sphingomyelins are also phospholipids without glycerol. Okay, so here, from here also you will get some question. Now, the role of phospholipids in plasma membrane. It is very important that the main role of phospholipid of the plasma membrane is to make the membrane as barrier between two aqueous compartments, right? This. But in the plasma membrane, in the phospholipid, you will find there are some cis double bond which helps them to form a kink. In this kinky structure are actually inhibit them to freeze or to saturate among together. So these hydrocarbon chains, in presence of freezing, they do not form a saturated hydrocarbon, and they do not form a rigid structure. They maintain the fluidity. You have to remember that they maintain the fluidity even in presence of freezing temperature. So now, other than phospholipid, there are major important are glycolipids and cholesterol. Now, what are the role of glycolipids? Glycolipids are mainly involved in the binding of the peripheral membrane proteins and in the signaling mechanism. And the cholesterol, their role is very important. Now, here you find in the animal cells you will find cholesterol, but in the plant cell there are no cholesterol. So the cholesterols will not form a membrane by itself. So what it does, it actually the hydrophilic head of the cholesterol actually interact with the phospholipid, and they help in maintaining the fluidity at higher temperature. But what happens, when there is huge freezing temperature, when there is freezing temperature comes, like cold condition comes, this cholesterol actually interfere in the hydrocarbon chains, in the binding of the hydrocarbon chains of the phospholipids, and they maintain the fluidity. So first they maintain the uh, uh, they inhibit the rigidity in the in the low temperature, and they maintain the they inhibit the rigidity and maintain the fluidity in lower temperature, and they inhibit the fluidity in higher temperature. So that this is a very important role. You can study here what I have already said. It is very important you study from this. You take a screenshot and study here that this role of cholesterol is very important. But the bacteria and the plants does not contain any cholesterol. The plant, instead of cholesterol, they contain some major long bulky sterol rings, and in the bacteria this role is played by the hydrocarbon chains of phospholipids. Okay. Now to prepare this slide, I have taken the pictures from the Bruce Alberts, which you can study as a reference, and from the cell biology of the Cooper. Thank you. If you like our video, please subscribe and comment and share with your friends to help them. In our subsequent videos, we'll show you some very good quality of material, and will be helpful for your study in the CSIR net exam. And after that, what we will do, we will whatever portions we are completing, we'll see what are the questions which are coming in the exam. So please like, comment and share if you like our videos. Thank you so much.